Picking right back up where we left off, we're going to talk about the ventricles. We're going to talk about the forebrain. We just recently discussed the dura mater and how that is a protective layer of the meninges protecting the brain and the skull area. So now let's talk about the ventricles. And so here there are some nice shots here of the lateral ventricle, which is here, and then you can see the third ventricle here, and this shot right here shows you what that looks like, and here, right here, and you can see the fourth ventricle in these shots. Really gives you a good idea of the scope of what these ventricles look like, and then the cerebral aqueduct here, and we'll talk about that more in just a second. But what I'd like to discuss is the fluid that is in the ventricles, as well as the fluid that's surrounding the areas of the meninges, as well as in between and around the cortex. And so the cerebral spinal fluid, which is kind of a salty liquid, fills these cavities and areas and serves a couple of different functions. So for one, it's a shock absorber, and so it's protective against the skull and the meninges and the brain. So if you were to hit your head, for example, it'll absorb some of that shock. It's also really important for transporting nutrients throughout the ventricles and throughout the brain, as well as potentially removing waste, which is something that we see a process occurring during sleep. Really important for cerebral spinal fluid to move that out. Another thing that I want to bring your attention to is this cerebral aqueduct. Now, this is a pretty narrow area. You can see here it kind of funnels down and bottlenecks some of that cerebral spinal fluid. Well, in a fetus that might, for example, have a blockage here at the aqueduct, or earlier into the ventricles, that's not good for the cerebral spinal fluid to flow, and it stops the flow, dramatically reduces it. What ends up happening is that cerebral spinal fluid gets sort of backed up, and the head of the fetus starts getting really much larger than it should for its developmental period. And so what a doctor might suspect, looking at an ultrasound with a fetus with a really large head, is, hey, we might have hydrocephalus. Now, hydrocephalus literally means water on the brain. What happens is that the head will get much bigger because it's actually filling with fluid instead of draining out in the aqueduct or in an area of the ventricle. And so this could be really problematic because this can cause too much pressure on the brain and can lead to deficits and things like that. So what will happen is a surgeon can actually create surgically enter in a shunt, and that shunt will drain the rest of the fluid into the abdominal cavity and basically in the peritoneal area, and will drain off that cerebral spinal fluid. So it needs to be able to drain, or you're going to see some complications. We can also see this happening in adulthood, of course, but this is something that is seen also in some children. So another thing that I want to cover is just the forebrain and what makes up the forebrain. So the forebrain's made up of the telencephalon and the diencephalon. So the telencephalon is that area, the large area of cortex in the two hemispheres that you typically see. Also the limbic system and basal ganglia. We're going to go into these individually. The diencephalon is made up of the thalamus and the hypothalamus. Now, when we look at the cerebral cortex, and remember we have two hemispheres, so we have two sets of these, it's broken up into four major lobes. And so the four lobes that we're going to talk about here, like the blue is the frontal lobe. It's important for making decisions. It's, arguably where your personality is housed and really complex thoughts. Also includes the motor cortex, which is really important here. The parietal lobe, which is more on the top of your head, is important for speech. It's important for somatosensory areas, so being able to feel your environment, know where you are in your environment, and also experience your environment in your perception. We also have in the back of our brain the occipital lobe, which is important for vision and visual processing. So this is an area that if you, for example, damage something on your eye, you might be able to repair that. But it's really difficult to fix things that are at the visual association area, or the processing center for vision, the occipital lobe. If you experience an issue with the processing center, that's a lot more difficult to fix, and your vision might be permanently damaged. Also, if you ever hit the back of your head and see stars, that's kind of like, you're kind of interrupting the visual processing center, and that's why you tend to have difficulty seeing stuff. And then we have the temporal lobe, which is right over the ears, important for hearing in the auditory association area, important for smell, this sort of region right here. And so that's kind of the cortex and what makes up the general sort of overview of the lobe. So we have the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And so this makes up the outer portion of your brain. And the sulci are all these little grooves right here, these little like valleys right here. And then the fissures are the really large grooves that are really easy to distinguish. So for example, these areas are really, when you look at the brain, you can really see some of these fissures really, really well because they're really deep grooves. And then we have the gyri, or the actual bulges. So here, these were like the bulge, like the mountains in the brain. And then we have gray matter and white matter. And looking at a side shot of this, it's a lot easier to see where the gray matter and the white matter kind of are separated. So I'm going to go back actually on a couple of slides just to kind of show you a little bit better on, oops, I thought this one right here was it. Okay, we're going to move forward and look at this one right here. I'm going to move forward just a slide just to show you guys what this looks like. So here's all the white matter here, okay? And then here's the gray matter here. And so if you look at what makes up white matter and what makes up gray matter, remember that white matter looks white. Okay, so it actually looks white, and that's because if I'm going to kind of remind you what the neuron looks like. Okay, so you have a cell body. It's like a motor neuron, for example. We have the dendrites. Okay, we have the nucleus in there. Then we have the axon, which is made up of myelin sheath. And if you guys remember, myelin sheath is made up of fat, and fat looks white, or whitish in a human brain. And then here's the terminal buttons here. So what we have is white matter being primarily made up of axons. So white matter is primarily axons. And gray matter is primarily cell bodies and connections. So if you look here, kind of think of it like here's all the cell bodies and here's all the axons, kind of moving through here and connecting to other areas. And so that might kind of help you remember that the gray matter is made up of the cell bodies, whereas the white matter is made up of axons, and it's white specifically because of the high fat makeup of those myelin sheaths. Then we have the corpus callosum, and that connects the two cerebral hemispheres, and we're going to go into that next. So here is just the cerebral, the corpus callosum is what binds together the two cerebral cortices, the two hemispheres. And we typically show it in a sagittal plane. And you can't really respect this or really have a good idea of how expansive the corpus callosum is when you look at it from this angle, because it looks, it's like, oh, okay, well, it's a structure, it's a band. But you can really appreciate it at the dorsal view and at the coronal view. So at the dorsal view, okay, these are these fibers that are extending out from one cortex to the next. It's a very rich set of connections. These are a bundle of fibers that are really, really well, they're tough and they're well connected. And here you can see, so this right here is this structure, right? And again, you can see these fibers really going all the way through the cortices. Here's a really nice sort of representation of what those bands look like. This is just a much nicer way to kind of appreciate these connections and how they look from hemisphere to hemisphere. And you can see just how important this these connections are to have a really integrated experience from one hemisphere to the next. We're also going to talk, so we talked about the four lobes, and I kind of touched on this already. So the frontal lobe is really important for cognition. It's really important for decision making, those higher level processing. Parietal lobe is important for your sensations because it has the somatosensory cortex. The occipital lobe, which is in the back of the brain, is really important for vision and visual processing. And the temporal lobe, which is located over your ears, is really important for things like hearing and taste and memory. The primary motor cortex is located in the frontal lobe, whereas the primary somatosensory cortex is located in the parietal lobe. And then we use this thing called the homunculus. And so what the homunculus kind of shows us is how these sensations are kind of moving throughout that we experience throughout the brain. So I've got two images here. So this one is the homunculus on this side, and the next slide is where you can actually see the strips from a different angle. So here we like to use the homunculus to demonstrate where the largest areas of sensory receptors are. Okay, so for example, there's a large portion of the cortex devoted to the hand, okay, and a really large area of the cortex devoted to the thumb. There's a large area of the cortex devoted to the tongue. And so if you bite your tongue, for example, this is something that you're going to experience an amount of pain or sensation in there. If you notice that, like the trunk, for example, and the elbow has smaller areas of the cortex that are associated with that, especially when you compare it to the size of the hand. And you can actually sort of demonstrate this. Something that is really fun to do in sensation and perception is you can take two needle points or pen points, and you can touch them at two different parts of your back, for example, and then see how far apart they need to be in order for you to be able to tell the difference. You can do this on the trunk and see just how far away those things have to be, and you can't really tell. They have to be pretty spread out for you to be able to really sense them and to tell the difference. Whereas, for example, the lips have a really a large area of the cortex devoted to them, and you just need a really small amount of space between those 2 points where you can actually tell the difference between those 2 points. And that's because there's more surface area of the cortex that's devoted to, for example, the lips. There's more surface area devoted to the thumb compared to the leg, compared to the back, things like that. And notice also that it's contralateral. So, for example, here on the right side, if you are talking about the right hemisphere, that's controlling the left side of the tongue. So just to kind of remind you that each hemisphere receives information from the opposite or contralateral side. And then here is just another sort of view of the primary motor cortex and the somatosensory cortex. Remember, so for each, the left and the right, you have a strip that goes along for each. And so what you might find is that, for example, if a person has damage specifically to like right here on their left hemisphere, you might find that they have difficulty moving their hands. They might have difficulty picking something up. They might have difficulty feeling. They might have lost some sensation in their hand and may not be able to use it like they normally would. Same thing, if you see some damage here, the person might experience some numbness in their face. They may not be able to move one side of their face, which is this kind of area here is really prone to stroke. And so there's some really important vasculature that runs through here where we typically see that a person might be experiencing similar symptoms if they experience an interruption of blood flow in that particular area. Here's another view of the lobes just to kind of show you how extensive they are, right? So, this is still the temporal lobe here. This is the occipital lobe, the parietal lobe here. Here's the frontal lobe. These are really good ways to kind of see how these areas overlap because you have the visual association cortex that's primarily in the occipital lobe, but it does go into some of the parietal lobe. The primary visual cortex is what's housed pretty solely in the occipital lobe, but you can see there's overlap. And that's important for, being able to have a nice plastic brain. You want functional plasticity as well as structural plasticity. You want the brain to be able to mold and change if it's damaged or if it's been exposed to certain kinds of experiences. And so that's all important. Some of those things to overlap specifically. So that's kind of how it's made up here. And and you guys can sit and look at all the different cortices and how they're broken up from lobe to lobe. So another important thing to structures to talk about, or systems I should say, one is the limbic system and the other is the basal ganglia. So the limbic system is made up of several different parts. The limbic cortex, it's made up, which is located kind of here. The hippocampus, which is right here, shaped kind of like a seahorse. We have the amygdala, and remember you have two amygdala, you have one on each side, so you have a left and a right, and then the fornix and the mammillary bodies, so the fornix being here, and then the mammillary bodies being here. All of these structures are really important for things like motivation, for emotion. For example, the amygdala is really important for emotion and for fear response. If you take a cat and you remove the bilateral amygdala, you'll actually see that the cat doesn't respond to scents, like, for example, a dog scent. It will no longer demonstrate fear. And this is not necessarily a good thing. People who have damage to the areas of the amygdala may make poor decisions because they're not considering certain aspects of the situation. The hippocampus is really close to the amygdala, and again, you have bilateral hippocampi, and so you have one on each side. Important for forming new memories. You guys might remember the case of HM. And so HM had his hippocampi removed due to severe epilepsy. And so this was kind of one of the areas where his epilepsy was starting. And so they wanted to remove those structures to stop his seizures. And what ended up happening was that he wasn't able to form new memories. So he only remembered everything up to his surgery. And then once you introduced yourself, he could remember you for the duration that you stood there and was with him, for the temporary being, for a short-term memory. But once he no longer engaged with you, then he would forget. And so of course we know that the hippocampus is really important for forming new memories, especially episodic memories. Turns out that it's not the sole place where we're forming new memories. For example, habitual or sort of nonverbal memory, non-declarative memory, is actually not necessarily, this structure is not necessarily necessary for that, because HM could in fact create new non-declarative memories. So we do know it's important for episodic memories, for episodes of your life, and forming new experiences that way. Now, the basal ganglia is really important for controlling movement. The basal ganglia is made up of the caudate nucleus, the globus pallidus, and the putamen. One really important thing to note about the basal ganglia is that the color of the area of the basal ganglia is actually darker. So if you were to take this, slice of the brain and check out the areas of the basal ganglia, what's really interesting is that it's made up of dopamine producing neurons, and so it should be darker in color. Now, individuals who have Parkinson's disease, those dopaminergic neurons are basically dying, and they're not producing dopamine like they should. And so when you see the autopsy of a person with Parkinson's, especially with really severe symptoms, the color is different. It's not dark, and it should be dark. And so, specifically looking at, like, for example, the substantia nigra. And so what's really interesting is that, so the substantia nigra is the area that we see being a darker color. and that is mostly associated with Parkinson's disease and the control of movement. So it's just a more specific area. When a person is experiencing Parkinson's disease, they have a problem with initiating movement. So this isn't that they can't move, it's just that when you go to take a step and you go to initiate that movement and to move your leg, individuals with Parkinson's disease have a lot of problems with that. We'll go into that in a much more detail much later in the semester, but I do want to kind of bring your attention to the fact that this is something that that is an important thing to consider. And very interesting that it was discovered and kind of put together when the doctor who was performing the autopsy discovered that this area was not colored correctly, and the person correspondingly had these issues, symptoms of not being able to initiate movement. So the Diencephalon, which is part of the forebrain, is made up of the thalamus and the hypothalamus. And these are structures that are really important for different things. So the thalamus itself is a sensory relay station. What it does is it filters through all of the information coming in and determines what things to pay attention to. So there's some theories that schizophrenia, for example, some types of schizophrenia, there might be an issue with the thalamus. So this relay station isn't working properly. And some of the sensory inputs that's coming in that should be ignored are actually attended to. So for example, we would call this maybe a hallucination. So if a person is experiencing some extrasensory information and it comes through, a person with a functioning thalamus might ignore that. And then a person who, for example, might have schizophrenia, that gets through the filter and they experience the sensation and perceive it as a real visual thing when really it's just a hallucination. Another thing, so that's one theory of what's going on, is the thalamus is not operating correctly in people with certain types of schizophrenia. Thalamus also is really important for sending projections throughout the cortex. So there's a lot of projections that are available through the thalamus. Now the hypothalamus, so here is the thalamus here. The hypothalamus is sitting right here. The hypothalamus is really important for a lot of really vital functions. The hypothalamus is made up of a series of nuclei, and those nuclei send out information and control a lot of different behavioral functions like eating behavior, being hungry, thirst, things like that. It's really important for regulating body temperature and making sure that your body's at the right temperature and you're not too cold or too warm. It's really important for the autonomic or the automatic nervous system. Really important for attachment and feelings of being attached, for example, to loved ones. Also important for things like sleep. So, for example, we'll talk about the suprachiasmatic nucleus, which is a part of the hypothalamus, which is important for circadian rhythm and alertness and feeling sleepy and all that. And so the hypothalamus controls the autonomic nervous system and also the endocrine system, which, remember, is really important for the pituitary gland and helps guide release of those hormones. And in the next series of slides for chapter 3, we talk about how hormones are related to development, and we won't go into specifically what part of the pituitary gland does what, but both the hypothalamus and pituitary gland are really important for releasing information and releasing those hormones. So this is just another view of what we were talking about. And again, just to kind of give you an idea that the hypothalamus, a lot of people think that the hypothalamus is just like a structure. It's made up of a series of nuclei, and it's really important to know that this is a lot of areas in the brain. It's not something that you can kind of dissect out. This is made up of a bunch of nuclei that have a lot of different functions. Also, this is the optic chiasm, and so this is important, for example, for vision, where the eyes are meeting. And above the optic chiasm, the suprachiasmatic nucleus, which is a part of the hypothalamic nuclei, is important for circadian rhythms and receiving inputs from the eyes to know what time of day is it, and is it time for us to be sleepy, time for us to be awake, etc., etc. So moving on to the midbrain. The midbrain is made up of the tectum and the tegmentum, as you can see here. And again, remember that these are a little bit more difficult from depending on the kinds of views that you're getting and the kind of cuts can be a little bit harder to identify. So the tectum, for example, is made up of the superior colliculi and the inferior colliculi. And so these are responsible for both vision and hearing. And so here we can see the superior colliculus. You have two, so it's the colliculi, important for vision. In this case, you can see where that's specifically located more towards the back there. And you can see the tegmentum is made up of the periaqueductal gray matter, which is really important for pain. And we'll see here, and we won't really go into detail here, but this particular area is a really important portion of the brain that's responsible for the sensation of pain and for controlling those particular experiences. And so if you go into neuroscience and you start to you dissect brains, or you're looking at the pain pathways, then you'll learn quite a bit about how these particular areas factor into the experience of pain. We also have in the tegmentum the reticular formation. And again, the reticular formation is just a series of cells. So like here's the pons, for example, and the brainstem here. The reticular formation goes down the brainstem here. And it is not a structure. It's just a series of neurons that kind of, and connections that run through that area. Really important for sleep-wake cycle, contain REM-on, REM-off cells that move you through REM sleep as you progress through those particular types of sleep at night. The red nucleus or red nuclei here, important for motor coordination. The substantia nigra, which we were, talked about earlier, runs through this as well and is important for reward and motivation. And as you can see here, is colored because these are dopamine producing neurons here. And just again, some more ways for you to kind of identify where this cut is coming from. So we have the thalamus here, and we have a slice right here. Okay, so this is all these structures that are located above the cerebellum. And so remember we have the cerebellum kind of being that whole structure right there. So moving on to the hindbrain, the hindbrain surrounds that fourth ventricle and is made up of the metencephalon and the myelencephalon. The metencephalon's got the pons and the cerebellum. The pons are really important for being able to relay information to the cerebellum. So, typically, you guys could probably locate the pons pretty well. Here, they look like that right there. And is really important for the cerebellum part and relaying information there. Now, the cerebellum, we often just attribute to just, movement and fine motor movements. There's a lot more that the cerebellum is responsible for that we're finding out. So we're finding more and more out about what the cerebellum is important for. But one thing that might help you remember is that so I'll tell you guys a story about one of my former advisors who was telling this story about a person that he knew who actually had damage to the cerebellum. So we're just going to kind of visualize it here. So this is the cerebellum, and so that particular person needed to ride the bus because this person wasn't able to drive, and so the person had to carry a card with them or a letter with them wherever they went. Every time they went on the bus, that basically was a letter from their doctor that said, "Look, I have damage to an area of my brain that makes me look like I'm drunk. And so the person kept getting kicked off the bus because they had damage, brain damage to the cerebellum. And what that translated to was symptoms. The person acted like and looked like they were drunk. So there was a bit of slurred speech, but more notably, the person's fine motor movements were slurred. They weren't able to walk in a straight line. It was very sloppy, and the person just came across as looking like they were intoxicated. And so one thing that we notice is when you consume alcohol, one of the areas that's impacted by alcohol is the cerebellum, which is important for movement and important for, basically being able to walk in a straight line in those fine motor movements, holding a pencil, being able to draw and write, things like that. But we're finding out that the cerebellum is becoming responsible for a lot more things than we typically originally thought. The final part, is the myelencephalon, which contains the medulla oblongata, which is really important, and we're going to point this out right here. So it's underneath the pons. Really important for vital functions like breathing. And these areas right here, okay, if you were to damage these areas, that's actually really, really problematic, because these structures here are so vital for breathing and heart rate and alertness, especially the reticular formation area, and that's important for consciousness. So if a person severs part of their brain, what they might actually experience is a coma that they never wake up from. They may never be able to be conscious again. And so conscious awareness is housed in this particular area here. So the final thing I just want to just to touch on is the spinal cord. And so if you take a more advanced neuroscience class, especially in graduate school, you're really interested in it, you'll need to know the nerves, and you'll need to know all about the fibers and the motor fibers and everything that's going through the spinal cord. I'm not going to get into all of that because I like to focus mostly on the brain in this class, and it is a more basic class. So we're not going to get into the spinal nerves. But I do want to kind of bring your attention to sort of how this information is sort of sent through the spinal cord. So we have the white matter and then the gray matter. Remember, these are all the connections up through to the brain. His is an example of a spinal nerve, and there's a whole set of those and how they relate to each part of the body. But just kind of, showing you the different parts here. The spinal cord's job is to distribute those motor fibers to the muscles and to the glands. And so we call this efferent. So it's away from the brain. It's moving those messages to the muscles and glands from the brain. And so this is kind of a mnemonic device. Some people like it, some people don't. It's up to you. I like the word efferent, and basically being moving the F away from the brain. That's kind of how I remember efferent, is moving away from. And then the spinal cord also receives messages, which is afferent, receives those somatosensory information that it's getting from different parts of the body. So the brain needs to have a two-way communication to both receive information as well as push out information, so that it can navigate the environment, basically, and experience the world around it in a sort of holistic way. So it's taking all this information from the fingertips, is taking information from the skin, from the eyes, and from the ears, and from, all of those things are working to paint the holistic picture of the experience. This final thing is just a silly picture, just to kind of bring it all back into the mapping of the cat brain. If you've ever had a cat, then some of these things might resonate with you. But if you have any questions, you can email me about any of the other structures that we talked about. And I do highly encourage you to use the Anatomy Atlas app to really explore these areas of the brain further, because you can actually identify structures and then move the brain around and see where those particular structures extend to. And it's a really nice picture that's a little bit easier visually than just a sort of a flat 2D picture. So I highly encourage you guys to just play with that app, just experiment, and get an idea of what the brain looks like sort of altogether.